Hello everyone. We will now move on to the next session in lipid metabolism. So today we are going to be discussing about ketone body synthesis and breakdown, and also the disorder that is associated with this. So which are the ketone bodies? There are three ketone bodies: acetone, acetoacetate and beta hydroxybutyrate. So as the name suggests, ketone bodies, that means they have a keto group, acetone, acetoacetate. But beta hydroxyacetate does not have a keto group, but it's still considered as a ketone body. These are water soluble lipids. So this is an exception from what these are water soluble compounds. So this is an exception from what we have seen. Fatty acids were not water soluble, so they required something for their transport. Lipoproteins are required, or fatty acids will bind with albumin. Cholesterol also cannot be transported through blood. It requires a transport compound like lipoproteins. Whereas for ketone bodies, they are soluble, so they can be directly transported in blood and do not require any carrier or a transport molecule. Usually when we use the term bodies, it refers to something that is not dissolved, that is generally particulate in nature, is what we call as bodies. So these are completely dissolved in blood. So again this becomes a misnomer, ketone bodies. As I mentioned during beta oxidation, only if beta oxidation is happening, then ketone bodies will be produced. Otherwise ketone bodies, if there is a defect or if beta oxidation is not functioning, then ketone bodies will not be synthesized. And these are mainly energy packets that are produced by the liver. They are energy rich. Acetoacetate and beta hydroxybutyrate are used for energy purposes, whereas acetone is going to be excreted as such and has no other function. So the primary ketone body is acetoacetate. This is the primary ketone body, and from acetoacetate we can synthesize acetone and beta hydroxybutyrate. Tissue where it is synthesized, exclusively synthesized in the liver. But you also need to remember that liver is one tissue which cannot utilize ketone bodies. So liver will only synthesize ketone bodies, but liver cannot utilize ketone bodies. Again, utilization of these ketone bodies is happening in the mitochondria. So RBCs also will be out of this. RBCs cannot utilize ketone bodies. The precursor for formation of ketone bodies is acetyl-CoA, and this acetyl-CoA is the end product of beta oxidation. So if you remember last thiolytic cleavage, we are going to get acetyl-CoA in every successive cycle during beta oxidation. So this acetyl-CoA is the starting molecule for ketone body synthesis. So let's look at the synthesis of ketone bodies. We have two molecules of acetyl-CoA which will condense together. Enzyme required is beta ketothiolase. This is a reversible reaction. So if you remember, beta ketothiolase is the last enzyme in beta oxidation, where it is responsible for the cleavage of the fatty acid to release acetyl-CoA. So the same enzyme is used here. So it can be used to combine the two acetyl-CoA molecules to form acetoacetyl-CoA, or it can be used for cleavage of acetoacetyl-CoA to give two acetyl-CoA molecules. So this is a reversible reaction. Next, acetoacetyl-CoA condenses with another molecule of acetyl-CoA to form HMG-CoA. HMG-CoA synthase is the enzyme, and this is a rate limiting enzyme. So now we have condensation of 3 acetyl-CoA molecules to form HMG-CoA ultimately. So one more thing you need to remember, these steps are common to both ketone body synthesis as well as synthesis of cholesterol. So synthesis of cholesterol also will start off in the same way, but the difference is cholesterol synthesis is happening in the cytosol, whereas ketone body synthesis is happening in the mitochondria. So you have the cytosolic and the mitochondrial isoenzymes that are present. So HMG-CoA now undergoes lyase. By a HMG-CoA lyase is now cleaved, and one acetyl-CoA molecule is released to give acetoacetate. So HMG-CoA breaks down to give acetoacetate. So this acetoacetate is the primary ketone body. So by a spontaneous decarboxylation, this carboxyl group is released as carbon dioxide to form acetone. So from acetoacetate, spontaneously acetone will be formed, and acetone is a volatile ketone body. Because it is volatile, it is going to be excreted through breath, and that is why when there is ketosis or ketonemia and acetone is being excreted, it gives a fruity odor to breath. The fruity odor is because of the because of acetone that is being excreted in large quantities. So acetoacetate has another fate apart from this. Instead, it can get reduced. Acetoacetate will undergo reduction. NADH and H plus is the coenzyme that is donating the reducing equivalents required. So NADH is converted to NAD, or it is oxidized to NAD, and acetoacetate is getting reduced to form beta hydroxybutyrate. So there is a hydroxyl group. The keto group is reduced and it becomes a hydroxyl group. So this is beta hydroxybutyrate. So acetone and beta hydroxybutyrate are the secondary ketone bodies. So because these are water soluble, they can diffuse into blood and are readily transported to all tissues and can be utilized for energy purposes. Normally, the amount of ketone bodies that are formed is very less, and the extrahepatic tissues will be able to utilize these ketone bodies, so there is no buildup of ketone bodies. But when the utilization is less than how much is being produced, that is when we land up with ketonemia. So as I mentioned, there are two types of HMG-CoA synthase. One is in cytosol for cholesterol and one is in mitochondria for ketone bodies. Now coming to utilization of ketone bodies. And for utilization, I have mentioned that liver cannot utilize ketone bodies. It's only going to synthesize the ketone bodies. Cardiac tissue, skeletal muscles, renal cortex make use of these ketone bodies for their energy purposes. Even brain will utilize ketone bodies to get almost about 75% of its energy. So for utilization, again it is a reversal of how synthesis happened, but one step is not required. Otherwise it is a reversal. So first, beta hydroxybutyrate is oxidized back to acetoacetate and NAD is getting reduced to NADH and H plus. So beta hydroxybutyrate is getting oxidized to form acetoacetate. This NADH and H plus will enter into electron transport chain to give 2.5 ATPs. Next step, the acetoacetate that is formed has to be activated. That is, it requires coenzyme A to be added. So the donor of this CoA group will be succinyl CoA. Succinyl-CoA will donate the coenzyme A group CoA to acetoacetate to form acetoacetyl-CoA, and succinate that is formed will go back into the TCA cycle. So, in TCA cycle normally succinyl-CoA to succinate we would have got one GTP. So, if succinyl-CoA is being used for this purpose, that GTP will not be produced. The enzyme required here is beta-keto-acyl-CoA transferase, or thiophorase is the older name for this enzyme. This particular enzyme is not present in liver. So, that is why liver cannot utilize ketone bodies. And the acetoacetyl-CoA that is formed is cleaved to give two acetyl-CoAs, and enzyme is beta-keto-thiolase. So, beta-hydroxybutyrate will form acetoacetate, which is converted to acetoacetyl-CoA and then cleaved to give two acetyl-CoAs. And the acetyl-CoA can be used for energy purposes. So, why are we synthesizing these ketone bodies? One, probably because they can be easily transported, do not require any carrier molecule for their transport. So, it is easy to transport them to all tissues, and especially useful when the body is in a fasting condition, so that the glucose that is there can be kept aside for tissues which exclusively require glucose for their energy needs. And in people who are having diabetes mellitus or those who are starving, there will be reduced supply of glucose within the cell, and therefore the increase in fatty acid oxidation leads to excessive production of acetyl-CoA, which in turn will lead to formation of ketone bodies. So, coming to disadvantages of forming ketone bodies, one is these are acids. Beta-hydroxybutyrate is butyric acid, acetoacetic acid. So, these are strong acids, so they can cause acidosis. When they are produced in excess, it is going to lead to metabolic acidosis. You will be studying more about this when you start the class for acid-base balance in the body. So, when ketone bodies are produced, leads to metabolic acidosis, that is lowering of blood pH. And when there is acidosis, there is impaired oxygenation of hemoglobin. So, oxygenation also will get affected. So, I think this is one point you need to remember, because one of the fads is the keto diets. So, lots of people are doing the keto diet where they are taking very little minimal carbohydrates and higher fats, so that you go in and only when you are forming these ketone bodies, it means that you are at that stage that is required for losing weight. Ketone bodies have to be formed in this keto diet. So, the disadvantage here would be it can lead to severe metabolic acidosis and impaired oxygenation. So, these 2 points have to be kept in mind when somebody is going on a keto diet. So, it would not be very advisable to start on a keto diet. So, coming to regulation. So, for regulation, we know that ketone bodies are going to be formed only when fatty acid oxidation, beta oxidation is happening. So, whatever factors are going to stimulate fatty acid oxidation will in turn stimulate ketogenesis. Whatever inhibits beta oxidation is going to inhibit ketogenesis also. So, if you look, if you remember what we discussed for regulation of beta oxidation, one was availability of these fatty acids, that is lipolysis. So, insulin is going to inhibit lipolysis. So, insulin will prevent ketogenesis, prevent lipolysis, whereas glucagon, epinephrine, cortisol, thyroid hormones, all these group of hormones via cyclic AMP are going to increase lipolysis. So, it can also lead to ketogenesis. Next is oxidation will happen only if fatty acids are transported into the mitochondria. So if the fatty acids are not transported, oxidation will not happen. So what is preventing this transport? It can be malonyl CoA that is formed during fatty acid synthesis that will inhibit the enzyme carnitine palmitoyl transferase and prevent fatty acids from being transported into the mitochondria. So when fatty acid synthesis is happening, ketogenesis will be inhibited. Now, when there is increased generation of NADH and oxaloacetate, so the beta oxidation will happen only when the blood glucose levels are low. So that means gluconeogenesis will be favored in this milieu. So when gluconeogenesis is happening, oxaloacetate is being converted to malate, and malate is going in for gluconeogenesis. So there will be depletion of oxaloacetate, and this in turn will inhibit the TCA cycle from functioning at its optimal. So when oxaloacetate is depleted, TCA cycle will be inhibited. So there is excessive accumulation of acetyl CoA, and this acetyl CoA will in turn be diverted towards formation of ketone bodies. So normally we produce very little ketone bodies, and that can be utilized by extrahepatic tissues. So therefore blood ketone body levels will be around 1 milligram per deciliter, and urine also will be the same. 1 milligram is excreted in 24 hours. So when the production far exceeds the utilization, that is when these levels increase in blood, and it is called as ketonemia. And when blood levels increase, it's going to be excreted in urine, and this is called as ketonuria, and the two together is called as ketosis. And ketosis will only be seen in two conditions, that is diabetes mellitus and starvation. These are the only two conditions, diabetes mellitus, starvation, where you see ketosis. So what is happening in diabetes or in starvation? So normally when glucagon is present, this acts at the receptor and then leads to lipolysis, release of free fatty acids, and this leads to beta oxidation, increase in acetyl CoA molecules which enter TCA cycle to give energy. So in diabetics, in diabetes mellitus, although glucose is present, it is not being transported into the cell. So within the cell there will be hypoglycemia, which leads to signaling, and gluconeogenesis will be happening. So all the oxaloacetate that is present is being diverted towards gluconeogenesis. So when the oxaloacetate is depleted, this is going to inhibit the TCA cycle. So when TCA cycle is inhibited, utilization of acetyl CoA will be hampered. So there will be build-up of acetyl CoA, and this acetyl CoA is going to be channeled towards formation of ketone bodies. So that is why in diabetics or in starvation there is excessive gluconeogenesis, and this in turn leads to ketone body formation. So what happens when there is ketosis? There will be loss of sodium and potassium and leads to metabolic acidosis. So the ketone bodies are being excreted in urine, they are anionic. So therefore there is displacement of bicarbonate, and sodium is taken away because sodium will form sodium bicarbonate and is being excreted in urine. So there is loss of sodium. Now there is increased urinary excretion of sodium. Now normally insulin will help to drive potassium back into the cell. So when in diabetes, when insulin is not active or there is no sensitivity to insulin, then there is potassium that is coming out of the cell in exchange for hydrogen when there is acidosis. So, you will be learning more about this when we do acid base balance. So, all you need to know is potassium is exiting from the cell and potassium is being excreted through urine. So, there is increased excretion of sodium and potassium. And because of the excessive glucose in diabetes that is there, that is being excreted in urine because it crosses the renal threshold. So, then this will lead to osmotic diuresis. So, there is increased urination leading in turn to dehydration. So, we have seen that there will be acidosis because these are acids and the bicarbonate is utilized for buffering these acids. There will be Kussmaul's respiration, that is, whenever there is acidosis, it stimulates the respiratory center and so there will be rapid breathing, there is a hyperventilation, this is called as Kussmaul's respiration. And also there will be a smell of acetone in the patient's breath, that is fruity odor in the breath as well as other body fluids. Urine also will have a fruity odor. And because of the osmotic diuresis, there is going to be dehydration. So, ultimately if this is untreated, it can lead to coma and even death if it is untreated. Now, how do we diagnose? If we are looking at the qualitative tests that can be done in the practical lab by students, then we have Rothera's test and Gerhardt's test for ketone bodies, presence of ketone bodies in urine. So, Rothera's, there is also a dipstick that is available and this method is what is being done in all the clinical labs to know whether ketone bodies are present or not. Quantitative, we do not estimate how much of ketone bodies are present in blood. As long as we know it is being excreted in urine, that is enough. So, we are not going to look at how much is present in blood. So, instead it is very important to monitor electrolyte levels, sodium and potassium, because I have told you they are going to be excreted. Look at the acid base parameters, what is the pH, how much PCO2 is there, how much PO2 is there. Acid base parameters have to be followed up. Now, if you want to differentiate whether the ketosis is because of diabetes or starvation, simple test would be look for blood glucose and urine glucose. If blood glucose is low, starvation. Blood glucose is very high, urine glucose is also present, then it is due to diabetes. So, treatment. We need to give IV insulin, maintain the fluid balance and continuous monitoring of electrolytes, because once we give insulin is going to drive back the potassium into the cell. So, from whatever hyperkalemia, increased kalemia in blood, it will now lead to a hypokalemia, which is very dangerous. So, you have to monitor potassium levels when insulin is being given. So, I guess that brings us to the end of ketone bodies. So, when you come for the session, please go through this or read from the textbook about synthesis of ketone bodies, about utilization of ketone bodies, which are the tissues that are utilizing, why are these ketone bodies formed, what is the basis for formation of these ketone bodies, and what are the signs and symptoms, and how do we treat the ketosis. Thank you.